Hello everyone, welcome to the series of GPAT MCQ. Today's topic for discussion is enzyme, subject biochemistry. This is Trupti Parab. Let's move on to the first question. Question: lysis belonging to the which class of enzyme as per International Union of Biochemistry system? Option A 2, option B 1, option C 4, option D 3. So the correct answer is option C 4. Enzymes are a protein molecule that help to speed up the metabolism or chemical reactions in human bodies. International Union of Biochemistry system classify enzyme into 6 classes based on their function. Then the order of numbering is fixed. So the class 1 indicate oxidoreductase, which is possible to carry the oxidation and reduction reaction. Class 2 indicates transferase, which will be responsible for transfer reactions. Class 3 hydrolase, class 4 lyase, class 5 isomerase, class 6 ligase, etc. According to the International Union of Biochemistry, the enzymes are divided into 6 functional classes and they are classified based on the type of reaction in which they are used to catalyze. The first class is oxidoreductase, which is responsible to carry out the oxidation and reduction reaction. Example is alcohol dehydrogenase, cytochrome oxidase, and so on. The second class is transferase. The examples are hexokinase, transaminase, transmethylase, etc. As you can notice in the picture, the group attached to A is getting transferred to B with the help of transferase enzyme. Third, hydrolase responsible for hydrolysis. The example is lipase, cholinesterase, etc. The fourth class is lysase, which is responsible for addition to elimination reaction. The examples are aldolase, fumarase, etc. The fifth class is isomerase, triose phosphate isomerase, retinol isomerase. As we know that the isomers the isomers are molecules or a compound with an identical molecular formula. So, to carry out the interconversion of isomerase enzymes are used. The sixth one is ligase, which are responsible for condensation reaction. And the ligase के example है glutamine synthetase, acetyl CoA carboxylase, etc. For one enzyme in each class, the systematic name along with EC number is given in the bracket. For example, the alcohol dehydrogenase having EC number 1.1.1. So EC number हमेशा नहीं पूछा जा सकता, but sometimes it can be asked in GPAT या NIPER examination. So try to remember EC numbers of enzymes mentioned in Satyanarayana. Satyanarayana is a reference book for biochemistry which is considered as Bible for biochemistry. Move on to the next question. Which of the following metal is attached to enolase? Option A Mn, option B Mg, option C Ca, option D Fe. That is manganese, magnesium, calcium, iron. So the correct answer is option B magnesium. Two categories of enzymes require metal for their activity or distinguish. The moral is certain enzymes require activation and here the metal act as a activator for that particular enzyme. So इसके दो types होते हैं, metal activated enzymes and metalloenzymes. So metal activated enzyme में क्या होता है, the metal is not tightly attached to the enzyme. So जो metal है वो exchange हो सकता है किसी दूसरे ion के साथ. So the example are ATPase having metal Mg2, that is magnesium, and calcium, while enolase having metal which is responsible for its activation is magnesium. While the metalloenzymes are enzymes which hold the metal tightly and they are not readily exchanged. Example: alcohol dehydrogenase, carbonic anhydrase, alkaline phosphatase, etc. require zinc. Phenol oxidase require copper. Pyruvate oxidase having manganese. Xanthine oxidase molybdenum, etc. and so on. So this enzyme and their metals can be asked in GPAT. Move on to the next question. Which of the following statement is true? Option A. Allopurinol is a non-competitive inhibitor of xanthine oxidase. Option B. Dihydrofolic acid is an inhibitor of dihydrofolate reductase. Option C. Epinephrine is a substrate for monoamine oxidase. Option D. Dicumarol is a substrate for vitamin K epoxide reductase. So the correct statement is epinephrine is a substrate for monoamine oxidase. So before understanding this table, remember that in biochemistry, the active site is the region of enzyme where substrate molecules bind and undergo the chemical reaction. So basically, every enzymes have their respective substrate. वो substrate उस enzyme पे जाके bind करते हैं and उनका एक chemical reaction proceed out करते हैं. So every enzyme and substrate has a significant role. And उस substrate को inhibit करने के लिए we have some competitive inhibitors which will inhibit the binding of substrate to the enzyme. Enzyme xanthine oxidase, the substrate is hypoxanthine or xanthine and the inhibitor is allopurinol. Significance of inhibitor: allopurinol is used in the control of gout to reduce the excessive production of uric acid from hypoxanthine. So hypoxanthine, when undergoes the reaction under the enzyme of xanthine oxidase, it produces certain amount of uric acid, and accumulation of uric acid causes gouty gout arthritis. To reduce the production of uric acid, allopurinol is a medicament which inhibit this process. So basically, allopurinol is used to treat gout disease and so on. Every enzyme has their unique substrate and unique inhibitor. The enzyme monoamine oxidase having substrate catecholamines. So catecholamines like epinephrine, norepinephrines, which are catecholamines having the catechol moiety, get degraded by the monoamine oxidase. As we know that the catecholamines is a compound in which the catechol moiety is attached with a NH2 moiety or NH group, and उस नाइट्रोजन का जब ऑक्सीडेशन होता है with the help of enzyme monoamine oxidase, it get degraded. So the inhibitors are ephedrine and amphetamine drugs which are used for elevating the catecholamine levels. As we know that the epinephrine or norepinephrines are hormones which are required for the smoothing function of human body, and these drugs are normally used to elevate the catecholamine levels. So when you try to read this chart in detail, you will understand that every enzyme has its significant substrate and significant inhibitor. When there is a malfunction in our body, malfunctioning takes place in our body, then we have to take medicament which will be helpful in achieving the therapeutic effectiveness, and some of them act by inhibiting the certain enzymes. Move on to the next question. The coastland model is also called as A. Substrate strain theory, B. Induced fit theory, C. Fisher template theory, D. Lock and key model. So the correct answer is coastland model is also known as induced fit theory. Fisher template theory is also known as lock and key model. It is very simple to understand these theories and models in which lock and key, like lock here will be enzyme and the key will be substrate. जैसे lock के अंदर हम key डाल के process करते हैं, in same way the enzyme will consider as a lock and the substrate will consider as a key. While the induced fit theory is known as Coastland model, the induced fit model is a model depicting the enzyme-substrate interaction. In this model, the substrate and the active site of enzyme undergoes changes to attain an optimal fit. सो बेसिकली यहाँ पे मॉडिफिकेशन होता है, मतलब वो फिटनेस इंड्यूस होगा जिस तरह से सबस्ट्रेट का स्ट्रक्चर है और एंजाइम का स्ट्रक्चर है. व्हेन दे विल ट्राई टू फॉर्म द एंजाइम substrate complex, there will be change which will attain the optimal fit and which will produce the enzyme substrate complex. Move on to the next question. What is the optimum pH of most of the enzymes from fungi? Option A 3 to 6, option B 4 to 6, option C 6 to 8, option D 2 to 5. So the correct answer is option B 4 to 6 pH is optimum for the enzymes obtained from fungi. All enzymes have an ideal pH value which is called optimal pH. Under the optimal pH condition, each enzyme shows the maximum activity. So, every type of enzyme has their own optimum pH, and at that pH you will get the maximum action. Most of the enzymes of higher organisms shows optimum activity around neutral pH 6 to 8. Enzyme acid phosphatase having the optimum pH 4 to 5, alkaline phosphatase having the pH 10 to 11, pepsine having the pH 1 to 2, enzymes from fungi and plants having the optimum pH to achieve the maximum action is 4 to 6. It is very important in GPAT and NIPER. This question can be asked. Which of the following is the oligomeric enzyme? Option A lactate dehydrogenase, option B pepsine, C ribonuclease, D glucanase and cellulose. So the correct answer is option A lactate dehydrogenase. The enzymes are classified based on their structure. The word monomeric enzyme is used if it is made up of single polypeptide. Example ribonuclease trypsin. Some of the enzymes which possess more than one polypeptide subunit chains are known as oligomeric enzymes. The example is lactate dehydrogenase, aspartate trans-carbon amylase, etc. While the glucanase and cellulose are hybrid enzymes. Move on to the next question. Which of the following scientists coined the term catalysis? Option A, Berzelius. Option B, Kuhne. Option C, Buckner. Option D, James Sumner. So the correct answer is option A. Berzelius coined the term catalysis. Kuhne used the word enzyme to indicate the catalysis taking place in the biological system. Buckner isolated the enzyme system from the cell-free extract of yeast. He named the active principle as enzymes, which could convert sugar to alcohol. James Sumner isolated and crystallized of the enzyme urease from the jack bean and identified it as a protein. So you have to remember this scientist and whatever term they coined or whatever discovery they invented, because this type of questions can be asked in GPAT. Move on to the next question. What is the optimum temperature of a urease? Option A 100 degrees Celsius, option B 70 degrees Celsius, option C 40 degrees Celsius, option D 60 degrees Celsius. So the correct answer is option D 60 degrees Celsius. It's an optimum temperature for urease. So for various enzymes, we have different optimum temperature similar to pH. Taq DNA polymerase and methyl adenylate kinase requires 100 degrees Celsius temperature. Optimum temperature for the most of the enzyme in the body is 40 degrees Celsius, while majority of the enzymes become inactivated at 70 degrees Celsius as enzymes get denatured as the temperature increases. Move on to the next question. Which of the following is true about Michaelis-Menten constant? A. Km or the Michaelis-Menten constant substrate concentration needed to produce full maximum velocity. B. A single Km is applicable for many enzymes. C. A low Km value means less affinity for the substrate. D. The Km value ranges from 10 raised to minus 5 to 10 raised to minus 2 moles. So the correct statement is option D. The Michaelis Menten constant value ranges from 10 raised to minus 5 to 10 raised to minus 2 moles, while the rest of the options are incorrect. Let's understand the other option as well. Km, or the Michaelis Menten constant, substrate concentration needed to produce half maximum velocity. Every enzyme has a different Km value. A low Km value means high affinity for the substrate. The Michaelis constant, or the Michaelis-Menten constant Km, defined as the concentration of a substrate that is transported at half of the maximum velocity of a transport. So basically कितना concentration substrate का transport हुआ है at half maximum velocity, ये हमें Michaelis-Menten constant बताता है. Move on to the last question. Which of the following is a commonly present amino acid in the active site? Option A, glutamine. Option B, serine. Option C, asparagine. Option D, tryptophan. So the correct answer is option B. Serine is the most commonly or frequently present amino acid in the active site. The commonly found amino acid at the active sites are serine, aspartate, histidine, cysteine, lysine, and so on. Among these amino acids, serine is the most frequently found. Thank you for your attention. Stay tuned for more such videos.